is Dr. Tony Jackson. I work in the biochemistry department at Cambridge University. And I'm a university senior lecturer in biochemistry. I'm involved in teaching and research in the university in the biochemistry and the MINS course. That's the medical sciences course. But we've been involved in developing or laying the foundation to develop simple, fast, lateral flow type assays to detect the presence of antibodies against the virus in patients. This would then be the basis of a cheap lateral flow assay that could be used to detect infection in Africa, in an African context. I think that's important. So the way we've done this is to exploit a technology called single chain antibody technology. And these are, so antibodies are, of course, the proteins of the immune system that the immune system generates against any antigen of interest, any infectious particle. And we can actually generate our own antibodies against any molecule that we're interested in. Quite a long time ago, actually, the techniques to generate what are called monoclonal antibodies were developed. But now we've been able to do is to take that technology a little bit further and to produce what are called single chain fragment antibodies. And these are small pared down versions of an antibody. So they're about a third the size of a normal antibody. And the advantage of them is that they can be made in bacteria, and we can actually make very, very large libraries, so of vast numbers of these single chain antibodies, which again are produced in bacteria. And we can use that to screen single chain antibodies to anything that we're interested in. And the things that we have been doing is to isolate these single chain antibodies to the human antibodies, both the IgG, which is the sort of type of antibody that you get after longer exposure to the virus, and also the so-called IgM class. And this is the first class of antibodies that you get when you're infected. So being able to distinguish IgM and IgG type antibodies can tell us whether you've just been infected or whether you've been infected for some time. The advantage of single chain antibodies is that you can make them in bacteria very simply. More difficult production of whole antibodies, IgG and IgM and so on, is usually more successful if you're working with mammalian cells, but that requires quite complicated fermentation systems and so on, whereas the single chain antibodies can be made in bacteria, as I say. I should also point out that another innovative thing that we've been exploring is the so-called capture antigens. So this is the thing that we hope to be able to detect people's antibodies binding to, you see what I mean? And this is, rather than make the spike protein, which many traditional lateral flow type assays that are used routinely in the West use, again, making the spike protein in bulk is difficult technically. We've gone for a technique where we've looked at all the different regions of the virus that the immune system consistently generates antibodies against. We basically put that together in what we call an artificial antigen. And that, again, is a protein that's very easy to make in bacteria. We can make large amounts of that very easily. So it's a question of manufacturing at scale cheaply using very simple, basic, well-established molecular biology techniques in bacteria. The applications are the core idea behind the test is absolutely general. You can use single chain antibodies against any antigen of interest, any pathogen or whatever. It's simply, you just simply go back to the single chain antibody library and rescreen against the protein of interest. And the technology that we hope to develop, I think, should be widely applicable to many of these other cases, whether it's detection of individuals who've been infected with malaria or whatever. I don't see why that technology cannot be applied again and again to many different conditions. What we do, and I keep saying this to my medical students, is that actually what we do is to understand the basic molecular nuts and bolts of systems, a huge foundation on which modern medical science is based. So I have been involved in a lot of protein chemistry work, biochemistry, cell biology, and so on. I'm not actually an expert in applying that in practical issues to do with clinical trials or anything like that. But that's one of the great things about the AfriDx, because we collaborate with many other groups who have all of the different experiences and can come together to generate these sort of synergy, if you like. So our contribution is really at the practical ends of making the stuff, putting it together, and hopefully getting an assay together, which can then be tested in bulk with clinical samples in the field.